Hello, everyone. Today, we're diving into a crash course on how cells harvest chemical energy.
We really are. We're talking about the fundamental engine that powers pretty much all life. If you've ever wondered how you turn, a sandwich into the energy to think or move, this is it.
It's all about cellular respiration, the process of converting food molecules like glucose into ATP.
The ATP, right? The cell's universal energy currency.
And you can see just how critical this whole system is from the very first moment of life. I mean, think about a newborn's first cry.
That first breath.
Exactly. That's not just a sound, it's this massive, immediate demand for a chemical that the whole process hinges on: oxygen.
Without that constant supply of oxygen, the most efficient part of this energy factory, the part we're going to get to, it just stalls almost instantly.
And the main point is to make ATP, that spendable energy. But the system also generates heat. And some cells have actually specialized to make heat the main event.
You're talking about brown fat cells. They are such a cool evolutionary adaptation.
I've heard they're important for babies.
Very important. Infants can't shiver effectively, so brown fat is their personal furnace. But adults have it too, and these cells run a special version of respiration. They have a kind of regulated short circuit.
A short circuit, how does that work?
Okay, so think of the main process as building up an electrical charge, like in a battery. That charge is then used to power a tiny spinning motor that makes ATP.
Right.
Brown fat cells have a special protein that basically pokes a hole in the battery. It lets the charge leak away before it ever reaches the motor.
So all that stored energy doesn't make ATP, it just escapes.
It escapes as pure heat. It uncouples the two processes. It's this incredible example of how the body can regulate and redirect this core energy pathway for a very specific need, like staying warm.
That's a great place to start. So let's zoom out a bit and, put all this in a planetary context.
Right, because our metabolism is just one half of a giant global cycle. On one side, you have photosynthesis.
Plants taking sunlight, carbon dioxide, and water to build sugar molecules.
And they release oxygen as their waste product. They create the fuel and the oxygen we need.
And then we come in and do the opposite.
We do the opposite. Cellular respiration takes that fuel, those organic molecules, and that oxygen, breaks them all down, and what do we release?
Carbon dioxide and water.
The very things plants need to start all over again. So energy from the sun flows through the system once and has lost its heat, but the atoms are just endlessly recycled.
A perfect loop. So let's look at the master equation for our side of that loop, using glucose as the fuel.
Okay, so you start with one molecule of glucose, that's C6H12O6.
Add 6 molecules of oxygen.
And the output is 6 molecules of carbon dioxide, 6 molecules of water, and then the big payoff. Around 32 ATP plus heat.
And that ties directly into breathing, right? You breathe in the oxygen.
To react it.
And you breathe out the carbon dioxide.
Waste product. But here's a little detail that trips people up. The oxygen atoms you just inhaled, they don't actually become part of the CO2 you exhale.
No. Where do they go?
They end up as part of the water molecules produced at the very end of the line. The carbon in your exhaled CO2 actually comes from the glucose, from the food you ate.
So it's a complete atomic shuffle. And that shuffling, that breaking down of glucose, is all driven by a type of reaction called a redox reaction.
Oxidation reduction. It's really a controlled form of burning. Oxidation is the loss of electrons.
So glucose is oxidized, it loses electrons.
Exactly. And reduction is the gain of electrons.
Yeah.
So oxygen at the end is reduced. It gains those electrons.
I heard it described as electrons falling down an energy staircase. If you just burn sugar, it's one huge fiery drop. All heat, no useful energy captured.
That's a perfect analogy. The cell is much smarter than that.
Yeah.
It doesn't let the electrons just fall. It uses these molecular shuttles, these little coenzymes, to catch the high-energy electrons as they're stripped from glucose.
And those shuttles are NAD+ and FAD.
Right. When they grab the electrons, they become NADH and FADH2. They're like little rechargeable batteries that are now fully charged.
So they're carrying all that potential energy.
All of it. And their whole job is to carry those high-energy electrons to the actual staircase, which is called the electron transport chain. That's where the cell will cache them in step by step.
Okay, so let's walk through that whole power plant. It has 3 main stages.
Stage 1, glycolysis. Happens out in the cytosol. Its job is just splitting glucose.
Stage 2, pyruvate oxidation and the citric acid cycle. That's inside the mitochondria.
And that stage finishes the job of breaking down the glucose.
And then the grand finale, stage 3, oxidative phosphorylation, which is where the real energy jackpot is.
That's where the turbines are, on the inner mitochondrial membrane.
All right, let's keep a running tally here as sort of energy scorecard. We're starting with 1 molecule of glucose. Our goal is about 32 ATP. Right now, we have 0.
Okay, let's start with stage 1, glycolysis. The name literally means sugar splitting.
And you said this is in the cytosol, so not even in the powerhouse part of the cell yet. And importantly, it doesn't need any oxygen.
No oxygen required. It's a series of about 9 steps, and it has 2 main phases. The first is called the energy investment phase.
You have to spend money to make money.
That's it exactly.
Yeah.
The cell has to put in 2 ATP molecules to get the process started. That energy makes the glucose unstable and ready to split.
Okay, so we're 2 ATP in the hole. Then what?
Then you hit the energy payoff phase. The split molecules are processed, and you get a return.
And the return is?
4 ATP molecules are generated. And we load up 2 of those NAD+ shuttles, turning them into 2 NADH.
And the ATP made here is through something called substrate-level phosphorylation. That's different from the main event later, right?
It is. It's the simple way. An enzyme literally just grabs a phosphate off one molecule and sticks it directly onto an ADP to make ATP. It's quick and dirty.
Okay, scorecard update after stage 1. We spent 2 ATP, made 4, so we have a net profit of 2 ATP.
And 2 charged NADH shuttles.
That's it. That seems like a pretty small return.
It is. And that's the key takeaway. After glycolysis, something like 90% of the original energy in that glucose molecule is still locked up in the 2 pyruvate molecules we just made. We've barely scratched the surface.
So we've just done the prep work. Now it's time to move into the mitochondria for stage 2.
Right. The 2 pyruvate molecules move into the mitochondrial matrix. But before they can enter the main cycle, they have to be groomed a little. It's a step called pyruvate oxidation.
A transition step. What happens here?
3 things happen to each pyruvate. First, a carbon atom is stripped off and leaves the CO2. That's the first bit of carbon dioxide we exhale.
Okay.
Second, we load up another NADH shuttle. And third, the remaining 2 carbon piece gets attached to a helper molecule called coenzyme A.
And that creates acetyl-CoA.
That's the ticket into the main event of stage 2, the citric acid cycle, or the Krebs cycle.
Right. So the acetyl-CoA hands off its 2-carbon group to a 4-carbon molecule already in the cycle.
Making a 6-carbon molecule, citrate, and then the cycle just spins.
And as it spins, it systematically breaks that molecule back down, eventually regenerating the 4-carbon starter molecule.
Exactly. Along the way, the last of the carbon atoms from our original glucose gets stripped off and released as CO2. But the main goal here is not actually to make ATP directly.
Wait, then what is the payoff for this whole complicated cycle?
The payoff is electrons. It is a massive electron harvest. For every turn of the cycle, we load up 3 NADH shuttles and 1 FADH2 shuttle.
And the cycle turns twice for every 1 glucose molecule, right? 1 for each pyruvate.
Correct. So from just stage 2, for 1 glucose, the total haul is 2 ATP, 6 NADH, and 2 FADH2.
Okay, let's update the scorecard. After 2 full stages, we have a grand total of 4 ATP. Still not much.
No, the pocket change is small.
But our shuttle inventory is huge. We have the 2 NADH from glycolysis, plus the 2 from pyruvate oxidation, plus the 6 from the cycle. That's 10 NADH, and 2 FADH2.
That's where all the energy is. We've successfully transferred almost all the potential energy from glucose onto those 12 shuttles now. It's time to go to the bank.
Stage 3, oxidative phosphorylation on the inner mitochondrial membrane.
This is where the electron transport chain is. A series of protein complexes embedded in that membrane. The NADH and FADH2 arrive and drop off their high-energy electrons.
And as the electrons get passed down the chain, like a bucket brigade.
The energy they release is used for one specific job, to pump hydrogen ions, which are just protons, from the matrix into that little space between the 2 mitochondrial membranes.
Building up a massive concentration gradient, creating a reservoir of potential energy, like that dam you mentioned.
An electrochemical dam.
Yeah.
And the whole thing is pulled along by the final electron acceptor waiting at the very end of the chain.
Oxygen.
Oxygen.
Oh.
It's so electronegative, it has such a strong pull that it literally sucks the spent low-energy electrons off the end of the chain.
So without oxygen to clear the way, the whole chain would back up.
It would jam completely. The
Oh.
proton pumping stops, ATP production stops. That's why oxygen is non-negotiable for this process. It combines with the electrons and some protons to form.
And now, the moment we've been waiting for, cashing in that proton dam. The process of chemiosmosis.
All those protons that were pumped into the inner membrane space now want to rush back into the matrix, down their concentration gradient. But they can only go through one channel.
The ATP synthase enzyme.
And this enzyme is a true molecular machine. It's a turbine. As the protons flow through it, they force a part of it to spin, like water turning a water wheel.
A literal rotor.
Yes. And that spinning, that mechanical energy, is used to smash a phosphate group onto ADP to create ATP over and over and over again at an incredible rate.
This is where almost all the energy comes from. So let's do the final tally. 1 molecule of glucose, what's the grand total?
The maximum theoretical yield is about 32 ATP. We got 4 from substrate-level phosphorylation earlier,
The pocket change.
and we get about 28 more from this massive oxidative phosphorylation engine. That's the aerobic jackpot.
It's an incredible amount of energy. But what happens if you can't get enough oxygen? You're sprinting for a bus, your muscles are screaming for ATP.
Then you have to switch to Plan D fermentation.
So this is the anaerobic option, no oxygen.
Right. And to be clear, fermentation itself doesn't produce any more ATP. The only ATP you get is the 2 net ATP from glycolysis, which, as we said, doesn't need oxygen.
So what's the point of fermentation then?
The point is to keep glycolysis running. Glycolysis needs a steady supply of empty NAD plus shuttles. If the main electron transport chain is shut down, the NADH shuttles have nowhere to drop off their electrons and get recycled.
So they get clogged up.
They get clogged up. Fermentation is an alternate pathway that takes the electrons from NADH and dumps them onto pyruvate, which turns the NADH back into NAD plus ryo. It frees up the shuttles so glycolysis can keep making that little bit of ATP.
And we see 2 main types. In our muscles, that's lactic acid fermentation.
Pyruvate becomes lactate. It's also what bacteria use to make yogurt and cheese.
And then there's alcohol fermentation, which yeast does.
Right. Pyruvate is converted to ethanol and CO2. The CO2 makes bread rise and beer bubbly.
You know, the fact that glycolysis is found in virtually every organism and doesn't need oxygen or these complex organelles, it really suggests it must be an incredibly ancient process.
Well, absolutely. It's thought to be a metabolic relic from before Earth's atmosphere had significant oxygen, billions of years ago.
Okay, finally, we should mention that this whole system is not just for glucose. It's a central metabolic hub for everything we eat.
That's a crucial point. It's incredibly versatile.
So how do things like fats and proteins plug in?
Well, carbs mostly enter right at the top, at glycolysis. Fats are broken down into glycerol, which can enter glycolysis, and fatty acids. And the fatty acids are chopped up into 2 carbon units that enter the system directly as acetyl-CoA.
Ah, so they bypass glycolysis entirely. Is that why fats have so much more energy per gram?
That's a huge part of it.
Right.
A long fatty acid chain is basically a string of dozens of acetyl-CoA molecules ready to be fed into the citric acid cycle. It generates a tidal wave of NADH and FADH2.
What about proteins?
Proteins are broken into amino acids. The cell deals with the nitrogen part, and the remaining carbon skeletons can be plugged into the pathway at several different points, depending on the amino acid.
And it all runs backwards, too, for building things. If you have too much energy, too much acetyl-CoA, the cell can pull it out of the cycle and use it to synthesize fatty acids for storage.
And the whole thing is beautifully regulated. If your ATP levels get high, the ATP itself actually binds to one of the early enzymes in glycolysis and slows it down.
Feedback inhibition.
Exactly. And if ADP levels rise, meaning you're using up energy, the ADP activates that same enzyme and tells the whole factory to speed up production. It's a perfect supply and demand system.
So that brings our deep dive to a close. The big ideas seem to be, it's a multi-stage factory, it works by the controlled transfer of electrons, oxygen is the key driver, and the massive payoff comes from that amazing ATP synthase turbine, about 32 ATP for every single molecule of glucose.
And if we circle all the way back to where we started, with that curious case of brown fat, the body has a built-in regulated way to uncouple that proton gradient from making ATP, just to make heat. So the provocative thought is this: if that process is chemically controllable, what's to stop us from developing therapies that could, say, activate that uncoupling protein in regular fat cells? Could we tell our bodies to simply burn off excess energy as heat, rather than storing it? It opens up this fascinating possibility of controlling our metabolism at its most fundamental switch.